Okay, I think we're going to get started. I see no more stragglers. And so what I will ask is if you could please turn your cell phones off. Simon. Okay, well, it's my pleasure to introduce one of the original members of the faculty on the school. Almost mid-successional, if you know about stand dynamics and how the history of the school has developed. This would be mid-successional. So Professor Berlyn, who is definitely, in terms of the lineage of the faculty, the person who has been on the faculty the longest period of time, since 1960. And he's still going strong, a real dynamo. And I don't know whether you know about the history of the school at that time, but it was very strongly post-World War II, industrial, little boxes being pumped out for suburbia, and Yale was doing its job of trying to figure out how to create houses. It was really big in wood utilization, wood technology. Greeley was abuzz with chip and saw and all sorts of wood technological instruments. And Graeme was hired on at that time because all these wood technology folks were thinking, my goodness, we know a lot about wood, but we don't quite know how trees grow. I'm obviously simplifying this. But at that time, Graeme was the first professor of wood anatomy and development. And he came doing his doctorate at Iowa State, fresh, having taught John Gordon. For those of you who know John, he was later our dean in the 1980s and 90s. And Graeme took on this sort of larger than role of teaching about the structure of wood, the cellular structure of wood anatomy, and how it develops in terms of trees. And that is what he is very well known for during his early period. And then later, I mean, as all faculty, and we look at all faculty, me included, and we do have a sort of successional development in terms of our sort of strengths and how we look at things. Graeme moved from understanding wood into understanding trees and forests, and actually back in the sort of middle 60s, he was one of the earliest professors who studied radiation in forests at the Brookhaven National Laboratory. So he was very well published back in that period on understanding how forests responded to radiation. If you think about the sort of the nuclear world and development of and the armament at that time, it was a very important research topic. And believe it or not, it came back to haunt him, because when Chernobyl happened, the Ukrainian government came to ask him to provide them all sorts of answers about what happens when forests get radiated. So he came out with a lot of formative publications on that, and then his next sort of, maybe I'm too long here, but here's a long story. Morphological change. I hope this is interesting. You didn't know, right, Jim? You didn't know about this. I didn't think so. Was tissue culture. And I don't know whether you guys remember about tissue culture, but this is when Gabal was just a young sprout looking at acid rain with Herb Borman and Bill Smith. And he's not going to talk anything about this. He's going to talk about biostimulants. Anyway, okay, well, so maybe I should wrap up quickly. But he did an enormous amount on tissue culture, formative work on tissue culture, before then moving into, all these cells that I'm trying to cultivate in a test tube. They need stimulants. And it sort of dawned on him since then to think about biostimulants, organic biostimulants, that aren't actual fertilizers, but that actually strengthen cell walls, for example. Vitamins. How does this actually stimulate growth? And so this is what we're going to talk about. And he has been on that trajectory for, I'm going to say, the last 25 years, give or take. Ever since I've known him, he's been on this biostimulant kick at a similar time as trying to understand leaf structure and development, which is another theme that he will not talk about. So, Graeme, it's a great pleasure. Oh, I do have to talk about his big textbook, which is the microtechnique, the histology of cell structure, the techniques of dissecting cells, and microtechniques of viewing cells under the microscope. He is sort of very well respected. His core authorship of that textbook got him to be the only non-medical person on the Biological Stains Commission, which is this national commission that oversees all histology. Everyone else is a doctor, in the medical profession, except for this lone, poor, unfortunate botanist. Anyway, Graeme, you're going to talk about biostimulants. We're going to hear all about biostimulants. It's a great pleasure having you on our faculty. And I just wanted to show you, he could talk about many things. Biostimulants is going to be the topic today. Thanks.
That was very nice of Mark. Of course, I've known him since he was a beginning master's student here. So I'm going to set the stage a little bit for why I decided to study these or why they needed to be studied. So let's talk about the Korzybski role. Alfred Korzybski was born in Poland. He was a Polish count, but he immigrated to the United States in the 1830s, get away from the Nazis, and then he founded the field of general semantics, first in Chicago, and then he moved to Lakeview, Connecticut, and he was a friend of G. Evelyn Hutchinson, and therefore he had a presence at Yale. He was gone before I arrived here, but I heard a lot about him. And he developed this concept, this horizontal concept of life. Plants he called the energy binders of the world, and they bind the energy of the sun into chemical bonds. Some of these chemical bonds we call food, others we call wood. But they also, in addition, remove CO2 from the atmosphere and add oxygen in the process of photosynthesis. I should say that they have been very successful. They have reduced CO2 to where it is now limiting for photosynthesis. And so a lot of people are studying and predicting horrible things are going to happen when the CO2 goes up. But the plants don't mind. They love it. They're going to survive us, but we may not survive them. So another seminal person, is, I go back, this animals, Korzybski called space binders because they can bind different areas of land into a habitat, so they bind space. They're also environmental binders because they can build dens and shelters for themselves. Humans, being animals, are also space binders, but the plants are the ultimate source of all food on this planet, either directly or indirectly. And with 10 billion people expected to populate the Earth, eventually I think food is going to be a priority, and so that's another reason for studying it. There are approximately 500,000, give or take 100,000, species of plants on Earth. About 20,000 are classified as useful plants, but only about a dozen species provide the vast proportion, around 90% of our food supply. So that makes our food supply very vulnerable. And in addition to a lot of people, we're also occupying a lot of the land with cities and housing and buildings that formerly were prime agricultural land. Iowa is a tremendous, terrible example of it. Des Moines, Iowa is on a flat plain. It also has hills around it, around the Iowa River and the Des Moines River. But some of the most productive farmland in the world is there, and it's now all houses. When I was a student at Iowa State in the 50s, we came and studied that Tama Muscatine soil that was there. So that's another reason food is going to be critical, and we need to develop sustainable ways to provide it for this huge population that we're expecting. Nobody has a crystal ball, but unless we have nuclear war or some catastrophe like that, it appears that we're heading in that direction. So another concept that I think we need to keep in mind is that of Vladimir Vernadsky, 1863 to 1945. Around Yale, everyone thinks that G. Evelyn Hutchinson was the founder of biogeochemistry, but actually Vernadsky was long before G. Evelyn and is considered elsewhere to be the founder of biogeochemistry. And he stated at the turn of the century that we're now in the third stage of the history of the Earth. The noosphere, which is dominated by human cognition. What he meant by that was human mental abilities to provide increased technology. And now people call that, we're now in the Anthropocene, but definitely dominated by humans, and food and renewable resources are keys to sustainability and survival. Well, now I'm going to just show you what these biostimulants can do. We don't just study a single crop on soil, we study a series of crops grown on soil to see their sustainability. So this is on your left are some radishes grown with the biostimulant. On the right are the controls that were grown with just mineral nutrients. And I should like to point out the people that were involved in these studies. I'm not going to give you a long list at the beginning because that doesn't really tell you who was involved with what. So as we go along, I will mention some of the students that were involved here. I think a lot of you still remember Achampong out of Boteng. He's from Ghana. He's back there now. And in addition, another person that worked on this was Chongrong Chan from Singapore, and also Charlie Ferris, who is now entering a master's student here. He worked with us for a couple of years as a tech before coming here as a student. So I hope that most of you know them. Incidentally, I asked my class if they'd ever heard of G. Evelyn Hutchinson, and not a single person had heard of him, including the TA. So for those of you that are a little older, you'll be amazed at that. But people pass and be forgotten, and that's, of course, what's happened to Vernadsky, so not many people remember him. But he was quite a brilliant scientist, and he had a lot of perspective on life and the biosphere. One of the reasons for the disparity of yield that I showed you is about half of all the inorganic fertilizer that you add to the soil immediately drains down into the water table, and the plants don't get it, but it contaminates the water table and can cause eutrophication. But the organic biostimulants have a much higher retention in the soil. Oh no. So this is a study with the organic biostimulant, and this is the leaking off, the runoff from the pots with a standard Doggett 20-20-20, and this is actually ammonium nitrate, and this is the control. Now, with these plants, for the first one, we were using a potting mixture that has slow-release fertilizer in it, so even the control was leaking phosphorus. Nitrogen, obviously the ammonium nitrate fertilizer at a huge peak, and this is the organic biostimulant, and this is the Doggett 20-20-20. So amazingly, the organic biostimulant holds on to the nitrogen because it has organic nitrogen in it in the form of amino acids, and it retains them in the soil, and it gets tied up both in the microbes but also by the roots and in the rhizosphere, and more of it goes to the plant. So on repeated cropping, you can eventually reduce the amount of fertilizer you have to add, well, the amount of organic fertilizer, because it stays in the soil. So if the plants don't use it all, and they don't, it still is available for the next crop. But the increased use of inorganic fertilizers has a lot of environmental consequences. And there was a study done by the Forest Service in 2015, and it indicated that 60% of reactive nitrogen came from nitrogen fertilizers that were applied in agricultural areas far from the winter mountains where the study was done. These are the winters here, and so a lot of it came from the atmosphere, so it was volatile because they use a lot of urea and ammonia fertilizers now, which volatilize and are then rained down hundreds of miles away from where they were applied. But of course, we're going to need food, so we have to have alternatives, because otherwise we're going to continue to increase the amount of inorganic fertilizers that use. So this is going to be a challenge, and we hope that plant biostimulants with their organic nitrogen can at least help to solve part of this problem. So here at FES, we develop plant biostimulants, and actually, by fortuitous situation, we actually got a company to produce it, and it was a very profitable company called Roots Inc. And I realized that the CEO, he was a very talented business person, and he was able to make it work. I would provide formulas, and then he would reduce them to make them economically feasible. But right now, I'm also trying to figure out the optimum, because the optimum may not be economic today, but in the future it may well be if this scenario that I'm presenting to you occurs. So with that company, we also produced a variety of products. It was about that, we started this, I started it in the 80s, but I sort of around 1985 is when I met up with Bob Weltzing, and the Roots Company was formed. But eventually we had mycorrhizal roots. People were not able to culture the spores. They're not able to produce the spores until about that time. And then Michael Amaranthus, who was at Oregon State working with the Forest Service at their cooperative experiment station, utilized that technique, and it seems once these things were developed, then the technology of doing it became available. So we had mycorrhizal roots, we had insect resisting roots. We had a student here, Bala Siva Ramakrishnan, known as Bala, and she worked with us on that formulation. Her husband's a professor in anthropology here. And we also produce gels for water stress. They can be very dangerous. They're available. They're polyacrylamide. Polyacrylamide's not toxic, but the monomer is very toxic. There was a plant physiologist from Australia, and I got a letter asking to send any audio tapes of physiological processes, because he was blind and paralyzed from a leak of the monomer. And so that's a little alarming. So we developed one with cellulose. It hasn't taken off, but cellulose is relatively non-toxic. It's all over the environment, and so that can be used. I don't know of anybody that's using it now, but hopefully in the future, because in arid areas this is a real problem, and it's going to get to be a worse problem with global warming. So if you believe in global warming, which I hope you all do. So our current product is modified from roots in that it contains more amino acids and no humic acids. We found that the organic nitrogen, as I said, is better retained in the soil, but also there was a big increase in the use of humic acids and biochar, which is sort of lightly cooked humic acids. It's really lightly cooked charcoal. Unfortunately, you have to use it at very low concentrations, and if you do continuous cropping, it builds up and becomes toxic. So I don't think we'll continue to hear about that as people have negative results over time. Just point out that I've looked at several of these graphs that are put out by government agencies and independents, and our inorganic fertilizer use is really increasing rapidly. So we really are going to have a problem with that. This is a model that I developed many years ago on fertilizer use, and this is based on Michaelis-Menten kinetics. If any of you had a course in biochemistry, you know that that's used to monitor the speed of enzymatic reactions, but you might not know that it's also used for mineral uptake. One of our students, Dave Ellum, used it as a photosynthesis model. He didn't develop that himself, but he found people that were doing it, and he used it very successfully. We had a systems ecologist here by the name of Ken Reed, and he was using it in forest growth models. So it was a very useful technique which was applied to many fields. It has a sad story. Maud Menten, who developed this technique along with Michaelis, and she was one of these people she was a Canadian, and she spent her whole life as an associate professor, I think primarily because she was a woman. Unfortunately, Bart Giamatti got rid of that procedure, and unfortunately it's being reinstituted again. Anyway, so this is an exponential curve, and what it shows is this is the amount of fertilizer in order to increase the yield. To go from 90% to 95% maximum yield requires 2.1-fold more fertilizer. To go from 95 to 99 requires 5.2-fold increase in fertilizer, I should say soil resources. So obviously it would be environmentally beneficial not to go for these extremely high yields, but we may not have the luxury to do that. And so trying to reduce the amount of inorganic fertilizer could be an important problem. So another thing about these biostimulants that we produce is that they don't have any hormones. There are several reasons for that. One is we're finding that microRNAs, for example, are used in human metabolism. So what you eat doesn't necessarily get completely digested. Some of the chemicals that you eat actually function in your metabolism. So we say that they accelerate growth and stress avoidance, but there's always a conflict between the two. So we can really increase growth, but that usually means at a sacrifice of stress resistance. We don't know that for a fact. Well, we partially know it for a fact because I did do some studies on it, but we need to do more. And it also alleviates stress. We try to make sure that these are all biologically safe. That's why I say we're not using the polyacrylamide gels anymore in the biostimulant, and we're not using the humic acids, although they're not toxic. They just build up in the soil, and they also tie up a lot of the nutrients. So we focus on antioxidant activity and insect deterrence. And my original design was if we could reduce inorganic fertilizer by 50%. Now I think we can do better than that, and maybe if we don't go for the 100% maximum yield, we can probably totally eliminate the inorganic fertilizers, particularly the ones that vaporize and can spread this contamination over hundreds of miles. Well, hopefully one picture is worth 1,000 words. Anyway, so you can see the control on the right and these various formulas of biostimulants on the left. And this is a product we were trying to develop called Shoots, which has arginine as well as the core vitamins and other biostimulants. But the organic nitrogen came from arginine, and these are various tests. This is a mixture of glycine and arginine, and this is arginine and salicylic acid, aspirin, and but now there are other plant stimulants on the market, and this is SN14, Neptune's Harvest. This is the control, and these are two of the biostimulants. This is the Shoots biostimulant we were developing, and this is the Shoots biostimulant with some salicylic acid and aspirin. Pardon?
You said that SN14 is another type of biostimulant?
Yes, it's not our biostimulant. It's just called SN14, unfortunately, so it may be confusing. It's not RS, which we've given up on anyway. But so we do pay some attention to cost now, so that I buy most of my chemicals from vitamin stores and bodybuilders supplement stores because people take this stuff. I've tested it out, didn't work out for me. So one of the things we've done is substitute glycine for arginine. And we found we can do that, not one for one, but glycine currently costs half the price of arginine. So we do take into account the cost. When we were trying to produce this with the Shoots company, and certainly with the Roots company, we had to take in a cost. But again, you can see there's a remarkable increase. And this just shows you, this is the control above ground vegetative growth of the foliage, and these are from the biostimulants. You can see it's about 3 times the biomass. And so these are 2 different biostimulants. On the left is glycine plus salicylic plus aspirin, and this is glycine at a slightly higher amount, which is now our standard amount, but also with beta-carotene. Beta-carotene is a carotenoid. The carotenoids protect plants from highlight stress and also from other stresses that we don't know about. There's a zeaxanthin cycle. Most of you don't know these cycles, but anyway, for light stress, but there's also other things that protect them. But again, at the current market value, it may not be economic. This is a study that was done by a young undergraduate, Alec Borsuk, and in 2016, or maybe it was 15. Anyway, he got 376% increase in cherry tomatoes produced with the biostimulant and 49% increase in radish biomass. Another thing that I took out of roots, which was an error, was adding some kelp. And for this study, we added kelp back in, and since Alec Borsuk was on the cross-country team, he got his teammates to do taste testing for us, and we found out that the source of the better taste was the kelp. So now we put the kelp back in. Actually, we had put it in before, but this confirmed that that was the source of the improved taste. Earlier we had done that with tomatoes at Greeley, and we had similar results. In fact, it was 100%. No one chose the control over the biostimulant-treated tomatoes. So the other thing that we found in a large study, which we did with a vitamin company, Hoffman Ranch at the time, we found that the vitamin B1 content in beans was directly proportional to the amount of B1 that we added in the biostimulant. So, I don't know, many have heard of yellow rice, which had vitamins in it, but I imagine it tasted bad. You don't hear about it anymore because B vitamins don't taste very good if you've ever tasted any of them. But anyway, so this the biostimulant beans had a lot of vitamin B1. That's not important for developed countries, but for many developing countries, that could be a factor in a way to introduce vitamins to the population. Speculation, but possible. Okay, so now stress can alter the composition of forest because species differ in their level of tolerance for a given stressor. I don't think I picked a good example for the current climate, but black spruce can survive minus 40 degrees centigrade. Bananas have died below 4 degrees centigrade. So it has a big, stress can have a big effect on the population of forests. But stress in trees, whether in parks or forests or city streets, can reduce growth. But generally, they don't kill the trees. So unless they kill the trees, people don't notice it. But Boyer was a very well-known soil scientist, probably dead now, but anyway, he found that his study claimed that stress applied to crop plants reduced yields by 78%. Well, how do the biostimulants work? Well, first of all, I'm not 100% sure of anything, but we know some of the things that they do. And I have to modify what I'm saying, that because no single formulation will be optimal under all conditions. So if you're working in the savannah in Ghana, then you may have to modify the biostimulant for that environment. So the formulas can be optimized for the particular environment and for the particular type of stress that's present. Just a little review. One of the critical stressors that we face, that the plants face, is reactive oxygen species. So you can get the superoxide ion by adding an electron to oxygen. You can get the peroxide ion, hydroxyl radical. And these form in large numbers in the leaves because in the process of photosynthesis, water is split and electrons are released, and they are used to form energy carriers, which are used to reduce carbon dioxide to sugar. And so you have all these quanta of light entering the leaf. You have these electrons flying around. The electrons can only pass down the photoelectron chain if each unit in the chain is oxidized in turn. But if they're reduced, then those electrons are not accepted and they're available to collide with oxygen. So reactive oxygen species are a critical stressor. These are free radicals or energetic molecules. And so I'm not going to say anything. So, but in face of all this potential destruction, chloroplasts and cells in general thrive and function. And this is in part due to the antioxidant pathways that we've discussed in C3 plants, which are the major type of photosynthesis. In fact, all plants do C3 photosynthesis. There are two other types of photosynthesis, but they're really just variations on C3. And so they have an organelle that usually is pressed to the chloroplast called the peroxisome, which has an enzyme in it, catalase, that can detoxify hydrogen peroxide. The chloroplast doesn't contain catalase itself, but it detoxifies the reactive oxygen species through ascorbic acid, superoxide dismutase, and glutathione. It forms a chain of reactions. And it does this, though, at the expense of NADPH, which is an energy-carrying compound, and so it's an energetic process. And for these reasons, the leaves of plants contain most of the plant's antioxidant constituents. They don't contain the vitamin Z, so you don't get colds. They are more selfish than that. They have the vitamin C to protect themselves from the reactive oxygen species. This is from a textbook, which I'm happy to see that the latest edition is Plant Physiology and Development. And these authors, who incidentally are all retired professors, have finally come to the realization that structure is important as well as function, and that the two are intertwined. So these are all the stressors that can yield to and cause reactive oxygen species, and they stop growth, development, and yield, and lead to stress responses. So the sources of stress, which are climatic that we talked about, edaphic, the soils, the physiography and topography, and the biotic, primarily competition, and then anthropogenic, sensu Vernadsky, but also sensu 21st century, where people are migrating in mass to the cities. Well, because of these factors, plants in the field are subject to multiple stresses, and they respond with stress physiology by producing more B vitamins, ascorbic acid, superoxide dismutase, catalase, and phytochematins and polyphenols and many other compounds. Now, if the plants were growing in ideal conditions with lots of water, lots of fertilizer, lots of light, not too much light, sufficient light, then things like biostimulants wouldn't be necessary. But in the field, they're never going into those optimum conditions, and that's why there's this big response to the biostimulants. Now, I'm not one of those people that condemn modern Western agriculture, because if that hadn't been developed by the state colleges through government supplementation, financing, millions of people would have starved. But we're confronting a different situation than we were in the 1890s, where there was lots of land, many fewer people, cities were much smaller, something like 80% of the population had rural origins, and so it was a very different situation, but I think we're facing something very different in the future. So the main ingredients of the plant biostimulant are vitamin C, that's the critical one. The amino acids, those are critical to provide organic nitrogen instead of inorganic nitrogen. Thiamine. Thiamine is amazing in that it promotes growth at low temperatures. And so it obviously protects from other stresses. And we did studies of this at low temperatures and even with a cryostat. But in combination with the other ingredients, it's even better at protecting from low temperatures. I thought I had discovered this effect, but then I was reading some of the research that was financed by the Work Progress Administration, by the Roosevelt administration. And there was a well-known plant physiologist that most have never heard of, named James Bonner, who was at Caltech, and he actually found this out with thiamine in the 30s. But in the paper he wrote on it, he said, well, it's a little significance because we don't grow plants at low temperature. Hello? We don't grow them. But how about Alberta, Canada? How about North Dakota? How about Siberia? And how about northern China? There are a lot of places where wheat is grown at low temperatures. It's been estimated if we could get wheat to grow at 2 degrees centigrade lower temperature, that would have vast increase in yields. So that may be an important factor also. So this just shows that biochar became toxic in the second harvest with radishes. This is earlier, woman named, that was Ray J. So's master's project here. She graduated in 2015. This was Vinland Bush, who was a postdoc with us for a couple of years. And this is the biostimulant with the biochar, but here is the control with the biochar. They were all given inorganic fertilizer, a small amount, 0.25 milligrams per liter. And this is roots with superoxide dismutase. But anyway, so it's not something that's sustainable, unfortunately. The other thing I'd like to point out is one of the reasons the plants benefit from the biostimulant is that the plants are interconnected. The roots are not separate from the branches, the stems, even though modelers like to model tree growth on the basis of branch growth. And when I was growing roots in tissue culture, they wouldn't grow unless you added B vitamins. And the reason is that the B vitamins are produced in the leaves and then transported down to the roots. So if you're going to grow roots in sterile culture, you have to add the B vitamins. So, and the same thing is another important hormone for plants is cytokinin, and the cytokinins are produced primarily in the roots and have to be transported up to the leaves where they're required for leaf growth. So they're all interconnected. Well, we've already talked about vitamin C. I'd just like to mention that Ricardo Russo was one of the important people that worked on that with me, was Ray Prendergast, who was a Yale College student and also an FES student. So this is a control in the cold. This wasn't really cold, but 15-degree days, 12-degree nights. This is thiamine in the cold, and this is the biostimulant that also contained thiamine, and that did even better than the thiamine alone. And of course, they all did well in the warm, but the roots in the cold did better than the control in the warm. So although the increased nutrient uptake and efficiency might make these plants more susceptible to insects, it turned out that the opposite was the case. And so Siva Christian did a dissertation on this. She was interested in entomology at the time, but got interested in using the biostimulants on the plants. Now, so other studies have since corroborated that vitamin C and E are active in insect detergents. And so some guy out west threatened to sue us for using vitamin E and C in the biostimulant, but fortunately we had a patent that was already over 10 years old before he even thought of it. So that ended that potential lawsuit. And one thing they do is, in addition to just producing protective compounds, when you treat the plants with the biostimulant, it makes the leaves tougher, and that makes it more difficult for the insects to attack them. And so this is plants exposed to hemlock woolly adelgid sugae, and this were the roots. So many fewer, less than half of the roots of the insects perform the biostimulant treated plants, hemlock seedlings, as opposed to the control. This is gypsy moth, and if the control was fed, it didn't really respond that well by producing more polyphenols. But when the roots were fed, they produced significantly more polyphenols. And the good thing is, if they were unfed, they went back to the control amount, because polyphenols themselves can interfere with growth. So there's pretty good control of this. But here's the leaf toughness experiments here, the roots in control. And Alex Finkrell, as well as Bala, were very important in designing the device that we use to measure leaf toughness. Now we have a fancy new electronic one, wherever it is. And, but the Yale machine shop in Gibbs' lab, no longer in existence, was able to manufacture this for us. So then when we built this, we got this new electronic one, we had them build, and the same machinist was still there, so he built the second one, and that was also used by Dylan Craven, and Gillian Bloomfield. Well, I'll give you one more concept. I realize people have class. In a number of species we studied coffee, we studied alders, we studied birches, pines, grasses, beans, soybeans, tomatoes, cherry tomatoes, and many other plants. And we've had positive results. One thing they do is they produce greater numbers of leaves at a given time. And this is called increasing plasticrons. And plasticron was a concept developed by Ashkenazi in 1880, and it's the time between successive leaf formation. And the benefit of using these kinds of measures of plant development is that the plants are more keyed to the plasticron than they are to chronological time. So we try to keep that on a biological basis. Anyway, thank you all for your attention. Sorry I went over, but...
First of all, I want to publicly thank Graeme. Graeme built a fluorescent microscope for my thesis, so back in the dark ages. But Graeme, thank you for helping me in your old dark room, your old microscope room.
Yes.
What led you to vitamin C? I might have missed that, but that seems to be sort of a critical part of your story, but I wasn't quite sure. Was that from the literature, or did you try that by trial and error? Did you get that from human vitamin C studies? Where did the vitamin C pop into your head or pop into the culture medium?
Linus Pauling.
Oh, and nobody.
I read Linus Pauling's book on vitamin C, and I said, hey, if this works so well on people, how about plants? Because I knew that they produced.
Thanks, Graeme. I'm curious why you tested your stimulants on radishes, or maybe that's primarily what you showed us. I would have thought that it would have been interesting to test them on other staple crops that are more consumed a little bit more than radishes, like maybe corn or wheat or potatoes. Why
It's
radishes?
only because I can do an experiment a month. They're like my fruit flies or Arabidopsis thaliana now of the plant molecular biologist. 30 to 40 days, we get a new crop. And so it's strictly an expedient, and it seems to mirror what we found in all these other species we use, but we'd only get one crop a year or two crops a year. This way we can do it. And then especially when we want to do repeated cropping on the same soil, every 30 days we can do that.
Graeme, you referred briefly to the cost of some of the raw ingredients that go into the biostimulants. I'm wondering if you've done a more extensive analysis. Does it pay off in the end to use the biostimulant in terms of the...
Well, it did with the Roots Company. They were a profitable company, and it also decreases transplant damage. In fact, there used to be some of the arboriculturists here told me they wouldn't put a plant in the ground without first treating the root ball with roots. So
on the same lines, what are the most common applications of your biostimulants? For example, when you are talking about the food problem to feed 10 million people of the world, how large, how extensive is the use of the biostimulants in agriculture? Is it only restricted to, say, greenhouses, or I think they have also been used for turf grass?
oh yeah, no,
But that's not food. So
it's not food.
I'm wondering how have you contributed with the use of the biostimulants?
Well, most of what we've done with food has been in small scale people. Michael Sterner, for example, did it in Africa, got a tremendous increase in yield. Some renewable businessmen bought thousands of dollars worth of roots, and they used it, but now it's not available. I got a letter from Vietnam a couple of years ago thanking me for my contributions to Vietnamese agriculture. I had no idea that it had even been used in Vietnam. But so I don't know, but certainly high valued uses. Like playing fields, was used in Wembley and several other British stadiums. I don't know if Wembley, but several British stadiums. Bob Wilson isn't here, so he would know, because he took care of most of those foreign contacts. And it's used for high-value crops. It works very well on orchids, for example. And so I think that the Roots Company then focused on turf and other products. It was used extensively in Japan, but again, that's very high level for golf courses. Toyo Green is a big golf course company in Japan, and they bought a lot of the Roots product, because the problem in golf courses is that the dead roots build up, and it's called thatch. But if you use the biostimulant, it promoted the microorganisms, and the thatch went away. And so they used that. So, but again, it's mostly been for the high value. To try to do it, we just never, we don't have the facilities to grow corn and wheat and things like that here. So we haven't seen what it could do there.
Okay, well, we have a class that's trying to get in here, but let's take this one last opportunity to thank Graeme.
Thank you. Okay. Hey. Nice to meet you.
I was looking at the stresses that you listed there. What are your views on latitude, altitude, and gravity? And are there any specific recommendations that you have?
Well, as a matter of fact, I can get moving on stuff. We've done extensive studies on altitude.
Does that qualify as a stress?
Oh yes, no. In fact, it's like a natural global warming experiment in reverse. Because as you go up, it gets colder. If you look at the reverse, it's coming down. So yeah, I had a student, Andrew Richardson. He did his thesis. Several of my students have done theses on altitude.
Not permanently.
And we also do latitude.
On a longer basis. On eBay. Well, there you go.
Okay, I will be there shortly.
Graeme?
How you doing, man?
Getting by.
You had a good break?
Yeah.
When's soup day? I was thinking about that the other day.
I don't know. I need to harass them because I would like some soup.
Yeah. I make the chowder for my family now, and they've basically just gone to whole cream.
It's the way to go. I
It
mean,
used to be half and half. Then it was light cream.
look, full fat's the way to go. That's where all the flavor is. May not be the best thing for you, but
Yeah, I
it's
know.
delicious.
I know.